Good morning again, grade 9, my advisory. Let's continue with the discussion. We actually stopped in the number of chromosomes in a human being. Okay, the number of chromosomes in a human being. When you say number of chromosomes, the total number of chromosomes, this is the totality. How many chromosomes do we have all in all? How many chromosomes do we have all in all? 44?
44.
Any other answer?
46.
46. It is 46 because it is 23 pairs. Okay? Because it is 46, 44 of it or 22 pairs of it is considered autosomes. Again, your autosomes are your characteristics, your attributes, your appearance. It is actually controlled by the autosomes. The 23 pairs or 44 chromosomes. Okay? Controlled by the autosomes. The 23rd pair, okay, the last pair or the two chromosomes on the last is considered as a gonosome or a sex-determining chromosome. Okay, it could be XX or XY. XX means female, okay, XY means male, right? This is also the reason why some of the characteristics are sex-driven. Like for example, when you say sex-driven, it can only be manifested in either male or either female. Okay, because of this. This is the reason behind. The egg always comes with an X chromosome. Okay, again, the egg is all carries or always comes with an X chromosome, while the sperm carries both X and Y. Okay, an X-bearing sperm fertilizes an egg. The resulting offspring is female because, again, the egg is always carrying an X chromosome. So if it is fertilized by the sperm carrying X also, it will result to a female or XX. But if a Y-bearing sperm, okay, this is sperm fertilizes an egg, the result offspring is male because it will result to X and Y. In short, the 44 chromosomes are autosomes, and the 45th and 46th, or the two last chromosomes, are the sex chromosomes or gonosomes. I'll put it in your chat box. Gonosomes. So, chromosomes contain 8,000 genes, while the smallest chromosome, the 21, the 21st, contains 300 genes. Okay. Let me check on the chat box. Gonosomes. Chromosome structure. Okay. Chromosomes are organized structures located in the nucleus of the cell. So because they are really small and they are really transparent, it is really hard to identify. But because of the microscopy, the advancement of technology, we can now actually observe this. They carry information essential to cellular functions such as cell division and cell differentiation. This is cell division. This is how we grow. We grow through cell division. Our cells divide by itself. Okay, through the process of cell division. It's either mitosis or meiosis. And of course, cell differentiation. When you say cell differentiation, like for example, the stem cell. Are you familiar with stem cell? Stem cell, everybody? Have you heard about stem cell?
No.
Have you heard about
Yes.
stem cell therapy? Okay.
No.
This is not common in the Philippines, but in the other countries, they're using stem cell therapy. Sure, what's with the stem cell therapy? What's with stem cells? Okay, stem cells are cells that are actively dividing. Okay, because they're actively dividing, they are tend to repair other cells. They are used in cell repair. Okay. What is cell differentiation? When we say cell differentiation, it came from a single cell, okay, and evolves to the different cells. Like for example, from zygote, we cannot actually identify what cell created the cardiac cell or cardiac muscle. What cell created the muscle cells? What cell created um, the cell of the Langerhans or your cells in your liver? Okay, so meaning to say, this came from a single cell, okay, and had undergone the cell differentiation. So the cell differentiation is the process of creating different cells out of a single cell. So that is cell differentiation. Okay. The term chromosome comes from the Greek word chroma, which means color. Chroma, okay, which means color, and soma, which means somes or soma, which means body. Okay. Chromosomes are called such because they're strongly stained by some colorful dyes used in research, because they are highly what transparent. We cannot actually observe them. We cannot actually observe them without dyeing or without staining or without putting some colors on them. Because of these colors, we can actually observe them. But if you're going to look at them in the microscope, even though you're using an electron microscope, those are the microscope used to observe those highly microscopic specimens. But then again, you just you will just see in the microscope a clear image, a clear specimen. There's no, you cannot actually identify them there without staining. So they need to be stained in order for us to identify. That's the reason behind. Chromosomes appear double-stranded in the nucleus of the cell before cell division. Okay, they are double-stranded. Okay, they are connected with another base, nitrogenous base. Each strand is called chromatid. Okay, and is made up of a long DNA molecule coiled around a protein called histones. Okay, the DNA molecule wrapped around protein core called nucleosome. Let's discuss about this later. I have an illustration for this in order for you to fully understand the topic. The nucleosome is the basic unit of chromosomes. Chromatids are linked at a point called centromere. This is the center of the chromosome. Okay, the bigger there is a bigger part of the chromosome and the smaller part of the chromosome, which divides the chromosomes into two sections. Okay, or arms. The short section, the smaller one of the chromosome, is called the P arm, and the long section or the bigger section is called the Q arm. Okay, this is now the illustration. This is the P arm, okay, and this is the Q arm, the bigger one. Okay, the smaller one is the P arm, the bigger one is the Q arm. Okay, at the center of them are called centromere. Okay, these are the centromere. And if you will remove one, this is the chromatids. We usually call them as sister chromatids. Okay, are you still with me? Please turn on your camera.
Yes, sir.
Okay, please ask questions in order for me also to address your concerns. Right. DNA structure and function. DNA is made up of many nucleotide molecules linked in a long chain. Okay. The basic unit of DNA is called nucleotide. Okay. Each nucleotide is made up of the following components. Okay. 5 carbon sugar. Okay. 5 carbon sugar called deoxyribose. This is a sugar. Okay. A phosphate group. Okay. Phosphorus. And a nitrogenous pair. The phosphate groups are actually in the sides. It holds the nitrogenous base pairs. Okay. Nitrogen-containing bases are grouped into purine or purine which include guanine, which is G. We usually use G in them, and adenine, which is A. And pyrimidines, which include cytosine, C, and thymine, T. Okay, again, guanine, G, adenine, A, that is under purine or purine, pyrimidines, which include cytosine C and thymine T. The DNA molecule is made up of the two complementary polynucleotides, or they are called complementary base pairs. Okay, they said to be complementary because the nitrogen-containing bases of one chain are bonded to those of the other chain according to the rule of base pairing. Okay, adenine A is always paired with thymine or T. A is to T, T is to A. Okay. Cytosine, which is C, is always paired with guanine, which is G. C is to G, G is to C. Okay. Hence, adenine and thymine are complementary bases, and guanine and cytosine are also complementary bases. So this is your DNA, as you can see. Okay. Guanine is being paired with cytosine, and adenine is being paired with thymine, and so on. So these are the complementary bases. Okay, complementary bases. And the phosphorus, okay, is on the side. This one, that which holds the DNA or the complementary bases, the nitrogen containing bases in place. Okay. Do you have any questions so far?
None, sir.
Question? All right, let's take it little by little. Please ask question. Gene structure. Okay, the gene consists of two polynucleotide chains. One of the chain is called the template, and the other chains is sequence of nucleotide or bases which help determine the kind of protein produced in a cell. This is actually one of the advancement of technology, simply because this is actually used by your vaccine. Okay, your vaccine is, like, for example, in an mRNA-based vaccine. I'm talking about the new vaccines that we have now, okay, from Pfizer, from AstraZeneca. They're using mRNA-based or messenger RNA. Messenger RNA, or ribonucleic acid, is a single strand. Okay, this is a single strand protein. Okay, since this is a single strand protein, okay, it carries a sequence of genes. Okay, and because this RNA is a single strand, it can be paired with our cells in our body. Okay, so tendency is it will create, or our body will create, a sequence of nucleotide or bases which help determine the kind of protein that our cell will produce in order for our immune system to react on it. That is the main reason. Okay, the mRNA base of your vaccines came from the spike proteins of the virus. So if you're going to look at the virus, I have seen this under the microscope. It has a lot of spikes. It looks like a tunok-tunok, no? But they are considered as spike proteins. So those spike proteins are actually removed in the process and create a vaccine out of it. Okay, there you go. The instruction needed to produce one particular amino acid. Amino acid is the basic unit of proteins. It's carried by a specific sequence of the nitrogen-containing bases known as codon. So, again, each codon determine a specific protein or amino acid that it carries. Okay. The information contained in genes are called genetic codes. Okay. Genetic codes are really long. That this is the reason also that the Human Genome Project takes a lot of time because they actually digest each genetic code. This is quite really long. Variation and inherited human traits. Okay, the combination of sex cells during fertilization results in variation in the offspring. What is variation then? Okay, let's take a look at variation later. This happens during the meiotic process or meiosis. Okay, this is the cell division again of haploid cells or gametes or germline cells or sex cells. During the first stage of meiosis, okay, crossing over takes meiotic place and allows the exchange of a part of chromosome with metaphase and other part of homologous chromosomes. Okay. It can occur in a kinetochore at a number of different points at the same chromosome. Meiotic microtubules, the microtubules, these are tubes that are micro. It actually attaches on the chromosome, okay, in the chromatids, or in the kinetochore, or in the centromere, and pull the chromosome away from each other. Okay, those are microtubules, resulting in the greater variation. Okay, until now, it is not being discovered what triggers the microtubules, what triggers the pulling away from each other of these microtubules. Okay, the points where chromosomes anaphase crossover is called the chiasmata or chiasma. Okay, variations also variations wait variations may also be caused by the permanent change in meiotic in DNA sequence called mutations. This we had discussed mutations last meeting, right? These changes may be in anaphase 2, brought about by exposure of ultraviolet rays, take note, or nuclear radiation or chemical, okay, or chemicals. It may also happen when cell replicates its DNA before it divides. So there are some lacking, okay, lacking DNA or lacking sequence of genetic code. When these mutations takes place in the gametes, they can be readily passed on. Most genes have two or more variations called alleles. Okay, for instance, the two alleles for hairline shape are straight and heart shape. Like in the widow's peak, this is a bird. Okay, a child may inherit some of the allele or two different alleles from his parents. Okay, the two different alleles will interact in a specific ways to cause trait to be expressed. Other inheritable traits, human traits, include the earlobe, our ears. Take a look at the form of our ears. Okay, my son has the same earlobe as me. Okay, it is really the same. Okay, because I actually pass on my genetic characteristic to my son. Okay, attachment, earlobe attachment, tongue rolling, eye color, the eye color also, the lip shape, the hair color, blood type, and hair texture. So these are some of the inheritable traits. What else? How about the formation of your face? The facial expression, those also can be, you can actually inherit them from your parents. Okay? The divided chin. Okay? These are some of the inheritable traits. The actual set of genes carried by an organism is called genotype. Again, the actual traits or set of genes carried by an organism is called genotypes. The expression or the station of an organism's genotype is called the phenotype. When you say expression, the one that you can actually observe, the one that you can actually see, that is actually the phenotype. The hair color, that if you can see the black hair, that's a genotype of particular, that is the phenotype of particular genes or genotype. Okay, what else? The color of the skin, I can see that the organism has a fair color, brown skin. So those are phenotypes. Again, the actual set of genes are called genotype. Okay, the actual genetic characteristics are called genotype, and the expression of the genotype is called the phenotype. Okay, the individual is said to be homozygous. Homozygous, homo means one. Okay, if the alleles of the trait are the same. Okay, if the alleles of the trait, for example, a person with two alleles for freckles is homozygous for that particular trait. Similarly, if the person has two alleles for no prickles, if the parent has a prickle and the other parent has no prickles, therefore he or she is a homozygous as well. Okay. Or similarly, if the person has two alleles for no prickles, if the both parents has no prickles at all, he or she is considered also as homozygous. But if one of the parent has prickles and the other has no prickles, therefore it is a mixture of two alleles. It is considered as heterozygous. Let's tackle heterozygous later. An individual is said to be heterozygous, again, when two inherited alleles for a particular trait are different, like what I had discussed a while ago. So the first parent or the other parent has a set of genes for freckles and the other has no freckles at all. So it has two different alleles. Okay. If it has two different alleles or two inherited alleles, two different inherited alleles, okay, it is considered as, or the organism is considered as heterozygous. Hetero means more, two or more. Okay. In this case, an individual who possesses one allele for freckles and one allele for no freckles is heterozygous. Correct. Alleles interact in either a dominant and recessive manner. Dominant and recessiveness. Okay. A dominant allele expresses itself and masks the effect of the other allele for the same trait. Like for example, a person has both allele for free or hanging earlobe and the allele for attached earlobe. However, free earlobe is expressed in his or her physical appearance because it is dominant over the attached earlobe. So it covers up again the recessive allele. So like for example, the color of the hair, okay, if you have a parent which is a male parent or your father has a black hair and your mother has a brown hair and the offsprings are all black hair, tendency is the genes or the allele for the brown hair is being covered up of the black hair. Okay, that is because the black hair there is dominant over the brown hair. So the process there becomes what? Becomes, or the concept there becomes now dominance and recessiveness. Okay, so the dominant allele or the dominant genes is being expressed while the recessive allele is being covered up by the dominant allele. Did you get the point? Did you understand?
Yes.
Any
Yes.
question?
Yes. None. None.
Right, right. The recessive allele is one of that is masked by the presence of the dominant allele, okay, and will only manifest if the organism is a homozygous for the recessive allele, or if during the cross, because we'll be using a Punnett square, we'll be crossbreeding, we'll be crossing off genes, okay. If during the cross, the mother and the father is carrying the same recessive allele, it can be expressed. But if there is a recessive allele, okay, it usually covers up. If there is a dominant allele, it is usually covered up. Okay, the recessive allele will be covered up always by the dominant allele. That's the concept of dominance and recessiveness. a trait is symbolized by one letter. For instance, an heirloom may be represented by the letter E, capital letter E. By the way, if it is dominant, okay, if it is dominant, or if the allele is homozygous, okay, it should be represented by capital letter E, or capital letters rather. It could be any letters of your choice. But in this case, we're using letter E. Okay. Its alleles may be written as the following manner. Okay. E. Okay. Because it is represented as E and this is a dominant allele. Okay. We have homozygous. Homo means one because it carries only capital letter E or dominant letter E. Correct? This is homozygous heirloom. Okay.